Hello, and welcome to our live webinar today, Next Gen Cytogenetics, Finding Hidden Clues to Health and Disease. Thank you for joining us. I'm Claire Rogers, Chief Marketing Officer at KromaTiD, and we're really looking forward to hearing from today's guest speaker, Susan Bailey of Colorado State University, who's live here with us in the Zoom room. And just wanted to give you, just before we get going with my introductions, just give you a little bit of housekeeping. You'll notice at the bottom of your screen, you have a Q&A icon, so you can submit questions. We will be able to see them on our end. All the other attendees will not be able to see them, but if you go ahead and fill it out and send it, we will see your question. And also, if you have anything to communicate to myself or the speakers, you can also use the chat. Any issues going on your end, please, let us know, and we'd be happy to address that. So to get started, Dr. Bailey is a professor of radiation and cancer biology in the Department of Environmental and Radiological Health Sciences here at Colorado State University, which is in Fort Collins. Dr. Bailey is a past president of the Radiation Research Society. She serves on numerous national and international committees and has been recognized as Innovator of the Year for her work on technology development and commercialization with KromaTiD. Susan is a native of Los Alamos, New Mexico, and received her Master of Science and doctorate degrees in biomedical sciences from the University of New Mexico School of Medicine. She's a radiation cytogeneticist with particular interests in telomeres and DNA repair, and her research efforts have resulted in over 100 peer-reviewed publications that most recently demonstrate the value of assessing telomere length, dynamics, and DNA damage responses as informative markers of life stress, aging, and radiation exposures in extreme environments. And it's been especially currently being applied under NASA's first one-year mission on board the International Space Station, and is one of the investigators in the NASA twin study. Before Susan starts with her presentation, I want to introduce our Chief Technology Officer, Chris Tompkins, who will say a few words to introduce the topic. Dr. Tompkins has more than 25 years of pharmaceutical and biotech industry experience, including executive roles at Avia Medical, Atrius Bioscience, and Prologo. He has been here at KromaTiD for the past decade, at least. And he holds a Bachelor of Science in Chemical Engineering from University of Michigan and a PhD in Chemical Engineering from North Carolina State University. And just one more housekeeping item. We are recording this, and we will be sending you a link to hear the on-demand version tomorrow. So take it away, Chris.
Thanks, Claire. And good morning to all of you. As Claire said, I'm Chris Tompkins, Chief Technology Officer here. And I'd like to, first of all, personally thank all of you for coming to today's seminar. Now, my role around here has been to take Susan's work and really make it practical and to commercialize it. And I'll be back after Susan has spoken to talk to you about how you can access all of the cool toys that we're really talking about today. Now, cytogenetics, the study of chromosomes, is really an old school science. G-banding, for instance, predates the human genome sequencing by about 5 decades. The main cytogenetic techniques, like FISH, have survived into the NGS era because they're clinically established, inherently single cell, and pretty importantly, orthogonal to any sequence-based method. However, these old school cytogenetics are fundamentally constrained by their underlying technologies, for instance, staining and back-derived probes. And so while robust and clinically important, they're all kind of stuck in the 1990s. Professor Bailey, more than any other individual, is responsible for bringing cytogenetics into the 21st century. By combining co-FISH with synthetic sequence-based probes, Professor Bailey was able to extend the range of cytogenetics to measure all classes of structural variants, while at the same time improving the resolution, sensitivity, and sensitivity, making a true next-gen cytogenetic technique, which we call DGH, or directional genomic hybridization. So now I'm going to pass it over to Dr. Bailey, and we'll be back at the end of the talk for questions.
Well, thank you, Chris, and thank you, Claire, to both of you for your very kind comments. It certainly has been a wonderful journey over the past number of years. I won't say how many. For me, it's been a while, but it is a pleasure to talk to you today about chromosomes. Certainly one of the passions that has driven my career. And I'd like to start by just a brief history. Chris mentioned some of these events, but just to really give you some perspective of where we are today. And to set the stage for that, just to remind you too, what chromosomes really are. In spite of the fact that they were first observed as in the 1880s, it was really only much more recently that we appreciated that chromosomes were made up of DNA, very long molecules of DNA, the genetic material of life that makes you who you are. And the DNA molecules are condensed and coiled down into very characteristic sizes and shapes of chromosomes that reside in the nucleus of the cells of your body. Chromosomes are made up of two sides or two chromatids, and they're held together by a primary constriction or centromere, and each chromatid or each end of the chromosome is capped by a telomere. It's interesting that the first accurate chromosome count of human chromosomes wasn't until 1956, and they established it at 46. Up until that point, they thought there were 48 chromosomes in the human karyotype. They then identified the 23 pairs and classified them into 7 groups. They also realized fairly early on that alterations in chromosomes were associated with disease. And some classic examples of that are trisomy 21, or 3 copies of chromosome 21 associated with Down syndrome. There's also in the 60s, the Philadelphia chromosome was identified by Janet Rowley, and that particular translocation between chromosomes 9 and 22 are associated with chronic myeloid leukemia. Of course, the discoveries that we've made along the way for chromosomes or in cytogenetics really have been driven by the technological developments that have allowed us to see them. And here you see that in the 50s, 60s, chromosome banding became a thing, and they were able to identify these light and dark bands along the arms of chromosomes. And they're just termed G bands for the Giemsa staining that was used to identify these bands. And they started classifying them then chromosomes, or identifying chromosomes based on their size, the position of the centromere, and then as well as their banding patterns. And there were a number of different banding techniques, G banding certainly being the traditional and it's still a standard banding technique used to identify chromosomes. There were a number of other techniques that developed along the way. My focus today will be primarily on the fluorescence-based techniques, in particular fluorescence in situ hybridization, or FISH, which came along in the 80s, which really did increase the resolution of identifying chromosomes and looking at different features of chromosomes. We'll also talk a little bit about the development of CoFISH, SCI or MFISH, and MBand, and then end up with DGH and how much of an improvement that is over our previous technologies. So just to give you a feel for what FISH is, or fluorescence in situ hybridization, it really is fairly simple and straightforward, and started off by just realizing that DNA is double-stranded, and if you denature it or separate those strands, you can hybridize double-stranded probes to it that you also denature. So it's just complementary probes hybridizing to its target DNA, and it has a fluorescent tag on it. And so you can then identify, for example, in this image of an interphase nucleus, you can identify fairly easily that there are 3 copies of chromosome 21 in this particular cell, and there's 2 copies of chromosome 18. So FISH was initially used then to identify numerical abnormalities. As the technology improved and we used things like microdissection to scrape off chromosome arms and generate probes, or flow cytometry, where we could isolate individual chromosomes, we were then able to come up with these whole chromosome paints, as you see here in the middle, and label them different colors. And that the example shown here just shows you a structural rearrangement or translocation between the two chromosomes, which suddenly started to be visible as we could look at these chromosomes and look at them in different colors. And then, of course, this really climaxed with the ability to color each of the chromosomes their own unique color, either by sky or MFISH, just different methods in order to do that. And that was really what opened the floodgates to show how very complicated some of the rearrangements between chromosomes can be. So lots of different translocations and dicentrics shown in the image on your right there. These are radiation induced, but again, it just really revealed how complex beyond what anybody imagined could happen was happening right before their eyes. The other thing that was really interesting, if you look at the interphase nucleus up in the left-hand corner here, that was painted again with the colors for the MFISH. You can tell that, in fact, chromosomes occupy very distinct domains or territories within the interphase nucleus. And that was another thing we didn't really realize until we were doing these fluorescence-based assays. So bottom line is that these technologies really did revolutionize cytogenetics or our understanding of chromosomes. But there still was a big gap, and that is that we're still not really able to tell much about what's going on within chromosomes. So the next step for fluorescence-based technology really was M-band, and M-band really did kind of combine the idea of G-banding with fluorescence. So they would scrape bands or microdissect bands and generate probes and label them with different colors. So you still ended up with a banding pattern. They're prettier perhaps, but they still have pretty poor resolution as far as identifying, say, an inversion or an inverted section of sequence within the chromosome. And it was very limited as well. You could only do a chromosome at a time. It was quite expensive, and you really needed a computer-assisted pattern recognition to identify rearrangements. So if we think about then where does this all leave us and how does it open the door to, next generation cytogenetics? I hope you've appreciated that. In fact, as Chris talked about, old school cytogenetics really did give us powerful tools to appreciate copy number variations and translocations and simple types of rearrangements between chromosomes. However, these techniques were all limited by low resolution and really not able to profile very complex types of rearrangements. So there were a lot of types, particularly rearrangements that occur within chromosomes, like inversions, that remained cryptic or invisible to our technology. And that's really where this next generation cytogenomics or directional genomic hybridization, or DGH, comes into the picture. And DGH really has its roots in a technique that was developed in the 1990s, which was just called chromosome orientation FISH or CoFISH. And it was named based on its ability to actually identify the orientation or directionality of particular sequences in the genome. It was used for a number of things in those early years, and it was very good with repetitive sequences. I'll show you examples with centromeres and telomeres and some of the applications that we were able to use it for. But it really did set the stage and open the door for thinking about strand-specific hybridization and how we could use that to really improve detection of rearrangements within chromosomes. So this is a picture out of the first publication of CoFISH, and it was really a very simple procedure. It's just based on growing cells in a single round of bromodeoxyuridine, which gets substituted into that newly replicated strand. You stain the slides with Herxt, which sensitizes that BRDU substituted DNA to UV light, which you expose the slides to and then digest with XO3. And all of that serves to remove those newly replicated strands. So what you end up with are parental strands and parental target DNA strands that are running in opposite directions, and they're single-stranded. So they're prime for hybridization of single-stranded probes. So now remember, I told you fish is all based on double-stranded DNA and double-stranded probes. Co-fish now is all based on single-stranded target DNA as well as single-stranded probes that have a fluorescent tag on them. And so centromeres, because they are made of repetitive DNA, so they have a repeat that you just design an oligo, a probe to, that then hybridizes in a number of spots. You can produce a signal at the centromere. And you can appreciate here that in this case, we hybridize a single-stranded DNA oligo tagged with a fluorescent probe to the centromeres of chromosome 18. And these, in fact, were the first single-sided or strand-specific signals that we were able to generate using co-fish. And of course, we were also very interested in telomeres, because telomeres are another example of a very repetitive sequence. They're at the ends of all eukaryotic chromosomes. And, you know, at the time, we knew that, for example, human telomeres were made up of the repeat TTA GGG. And that's an interesting story in itself. That was all based on fish. The sequence was identified based just really on trial and error of different fish probes until they came up with the right sequence that lit up the telomeres. Here you see that the first published picture of fish on human chromosomes with the telomeres lighting up. So we were able to use the telomere probe and come up with oligos that, in fact, you know, were very specific to either the G-rich or the C-rich telomere, and again, use our single-stranded strategy and light up. On the left here, you see mouse chromosomes with one of the telomeres in red and one in green. And on the right, you see human chromosome spreads with the same setup here. And you can really appreciate how very strand specific these probes are, and what pretty pictures you can also come up with. But this was a really beautiful example of the power of single-stranded hybridization strategies. We were able to use, in addition to just finding out orientation, because I mentioned that that's actually what co-fish was all about in the beginning. We didn't really know if these tandem repeats were all running in the same direction or not. But the fact that they actually did all light up on one side of the chromosome certainly told us that they were, in fact, all of the same orientation. They were tandem arrays of repeats. And in addition to that, we were able to get some structural and functional types of information as well. Just leading versus lagging strand telomeres, for example, on the left. And then also on your right here, you see another example of co-fish telomere signals. But in fact, in certain instances, you'd actually see an extra signal hop over to the other side. So we were able to pick up these recombination types of events that were occurring within telomeres themselves. So these came to be termed telomeric sister chromatid exchange, SC or SCEs, which are relatively common in normal cells all along the length of the chromosome, but they're telomeres and repetitive regions in general seem to be real hotspots for these recombination types of events. So we were actually able then to see an event that was happening within telomeres themselves within a single chromosome. So that brings us back to, well, what can single-stranded strategies, what can co-fish, the whole idea of that, do for us as far as looking at inversions or chromosome aberrations that actually result from double-strand breaks. So 2 double-strand breaks in a chromosome, as you see here, and that piece of that fragment of broken DNA actually popping out. And instead of going back in the way it should, it actually inverts and goes back into the chromosome. And that's essentially what is called an inversion. It's a complete reversal of the orientation of material within a chromosome. And they're very difficult to detect by any of the standard cytogenetic technologies that we have, because they often do not change the size or the shape of a chromosome. And so they're very difficult to detect. However, if we think about then, just historically, the problem has been then that we really don't appreciate inversion frequencies very well because we can't see them. So the idea was that we really don't know or didn't know, and I argue we still don't completely know how common inversions are. Although there was some evidence from early on that inversions should be much more frequent and be associated with a variety of different diseases, that we just weren't appreciating them because we couldn't detect them, that many of these inversions remained cryptic. But they were expected to have a lot of the same kinds of consequences for diseases as any other chromosome aberration like a translocation. So that brings us to, okay, how can we then think about using single-stranded strategies throughout the genome along the entire length of a chromosome and not just repetitive sequences? And how could we use that to help us see inversions better? So it really was only a matter of just kind of extending that idea of strand-specific hybridization, that you could then say, okay, well, we'll do the same thing. We'll grow cells for a single round in BRDU. We'll go through the photolysis and degradation, and we'll get down to our single-stranded parental DNA that is running in opposite directions that serves as our target. But what the real challenge with this is that now we need single-stranded probes to unique sequences all along the length of the chromosome. And that, of course, would be true for each and every chromosome, that if you were trying to look at the entire genome. So if this could be possible, that we had these unique sequences, then just as I just showed you, that in fact, when an inversion occurs and you get a reversal of this orientation for a fragment or piece of DNA within a chromosome, you would be able to detect it simply by a switch of that signal, a color switch over to the other side or to the other chromatid. So that's really where we started, and we tried lots of trial and error experiments, many of which didn't go very far. But the real key to making the bioinformatics of our probe design work was release of the sequence of the human genome. And once we had the sequence, then we could use much more intelligent type of design, probe design, to get at our unique sequences. Here you see the very first chromatid paint to chromosome number 3, which was designed on some of the very early builds of the human genome, and really just a brute force effort of targeting unique sequences, trying to be sure that they were of similar orientation. Again, a lot of trial and error there. The oligos had to be of similar lengths and melting temperatures so that they would work in the cocktail. And in fact, really just building these directional probe sets. And this first chromatid paint actually was composed of over 17,000 individual oligos. But as you can see on the inserts there, it really made a very strand-specific and beautiful chromatid paint. So the answer really to this problem of high resolution inversion detection and really discovery was in fact DGH, and this ability to paint individual chromatids. And then whether or not here on the bottom you see some of our earlier versions of the paint, and in fact after radiation, ionizing radiation exposure, which is very good at producing chromosome aberrations, including inversions, because it's inducing double strand breaks. You see this switch of the signal over to the opposite chromatid or other side, very, very easily to detect inversions. And really, the resolution of this has improved considerably to where we can detect really quite small inversions. And this one over here, the simulated inversion, is pretty interesting. That was just one we designed. We designed the probes of opposite orientation, and you can see again that you can really pick up a very small inversion with this technology. So one of the very first things we did to put DGH really to the test was to put it up against MFISH, for example. And here is our metaphases from a clone, a human clone that survived radiation exposure. And you can see that in our MFISH karyotype, we've identified a translocation between chromosomes 3 and 4. When we look on the right, this is DGH, the same clone. Looking again at, we see the translocation. You see it, the yellow arrow at the top, and I see I'm missing the bottom arrow, but on the bottom here, this is our translocation between 3 and 4. And then over on the left-hand side, you can see that, in fact, we also pick up an inversion, a switch of that signal over to the other side on chromosome 3. And that particular inversion, or any of the inversions that we saw in this clone with DGH, were completely invisible to the MFISH karyotypes that we did. We also reached out to Eli Williams and asked him to do the G banding on this clone. And again, he identified the 3-4 translocation. But again, you can appreciate how difficult the G banding is to pick up an inversion. He was not able to pick up the inversion. And then Marco Durante and his group at GSI did the M banding on this particular clone. And once again, there just really the resolution was not good enough with this technology either to pick up those inversions that we were seeing fairly readily with the DGH. So this really led us to also try, we were also in this kind of alongside of all this, also looking at next-gen sequencing. Could sequencing pick up any of these chromosome or structural variations? And in fact, it did a pretty good job of identifying the translocations, just like the others. But again, it was unsuccessful in identifying the inversions. So, again, I think this kind of sequence of events and technologies really pointed to the fact of the high resolution nature of DGH in identifying particularly small inversions. And certainly I'll also say that even since this study was done in 2018, that bioinformatics and sequencing has improved, but still struggles with identifying internal or rearrangements within chromosomes like inversions. And of course, DGH has also evolved and has improved over the years as well. Here on the right, you see, again, this same clone, our translocation between chromosome 3 and 4, as well as here we pick up a small inversion that's actually part of that translocated piece. So that's how we started chromosome 3, single chromosome coverage, single color. We then got a lot better at development of the probes and the chromatid paints, and now had chromatid paints or DGH paints to 3 different chromosomes 1, 2, and 3. And so again, you can see that really it enables this simultaneous detection of translocations. Here's another one between chromosomes. And at the same time, we pick up these rearrangements within chromosomes or inversions by just this single switch to the opposite chromatid. And then we see where we are today, which is just incredible, that in fact we can cover the entire genome, 5 colors, do the karyotypes, identify all of the chromosomes, and look for translocations, but particularly inversions. And you see some very nice ones here in chromosome 1, where the signal switches over to the other side, and also one here on chromosome 2. So just really, the technology has really advanced and enabled us to look at the entire genome, and do so with high resolution for both intra and interchromosomal rearrangements. And I've mentioned a couple of times to you that, in fact, radiation is very good at inducing double strand breaks, and that's why we see chromosome aberrations associated with exposure. But it's also true that with some of the modern editing strategies that we have now, they intentionally introduce double strand breaks into the genome. And so, chromosome rearrangements would also be expected as a result of genome editing. And in fact, that is very much what we see here, some very targeted DGH probes, so not whole chromosome paints, but targeted probes to specific sequences around edit points and edits that you can actually then look at color changes in color order and identify inversions that occur actually between edits, and then sometimes also even off target. So I'd like to go ahead and finish, spend a few minutes on some of the applications that we've used for DGH. We'll talk a little bit about biodosimetry, again, related to a variety of different radiation exposures. I'll also mention that DGH has been used to help improve diagnosis of rare diseases where they don't know what the disease is. Could it be potentially an underlying inversion contributing to the disease? We've also used DGH strategies in a number of aging studies and combining them with telomere as well as subtelomeric probes. Another important point is that, in fact, because our probe design is based on the sequence of an organism, we can also look in other species, any species really that has had their sequence, their genome sequenced, we can develop probes too. So here, this metaphase is actually from a wild boar in the Fukushima exclusion zone, where we developed some centromere probes to pigs or boar so that we could look at dicentric chromosomes. And my arrow again is a little bit off, but here's our dicentric chromosome. So again, it just illustrates that you can do some of these things, these similar strategies in any species. And we've done it here with boar, but also rodents and monkeys. And really, as I mentioned, any mammalian genome that's been sequenced. So let's talk a little bit about biodosimetry and radiation exposures. This was a study where we looked at US World War II veterans that were accidentally exposed to radioactive fallout during the 1950s associated with nuclear weapons testing. And I was just going to mention, if any of you have seen Oppenheimer, you realize that after the end of the war, certainly the Cold War began, and a lot of nuclear testing was occurring really around the world. These particular U.S. veterans were accidentally exposed. Many of them were weathermen that were exposed to a plume that the weather changed and the winds blew in an unexpected direction, and they were unexpectedly exposed to that radioactive fallout. There also were a number of individuals that were exposed at the Nevada test site during that period of time. And many of them received quite high doses, over 200 millisieverts. And what we were able to do in collaboration with Steve Simon at the NIH was to get peripheral blood collected from 12 exposed veterans and 12 age-matched veteran controls. So of course, these gentlemen were all over 80 years old. One was as old as 100 years old. So many, many years after the exposure event. And one of the first things that we did was we also needed to come up with a standard curve or a calibration curve. And we did that for inversions and using DGH. And we used 6 healthy male volunteers that were in their mid-20s, really to reflect the age of the veterans at the time that they were exposed. And this was the first time really that there had been a calibration curve for inversions done. And here you can see how their frequencies compared to those of translocations, both of course increase with increasing dose. But you can also appreciate that inversions occur even at a background level much higher than translocations, and that also they increase at a higher slope or a higher rate than translocations. So inversions then are more frequent than translocations, and they can be very informative because of that. We don't have to score near as many cells in order to get some significant results. So here I'm going to show you that, in fact, a few things we didn't know about inversions, and that is that age, their frequencies are influenced by age and smoking. So on the left, just like translocations, which had been reported to increase with age, when you see here in the red bar, and increased frequencies in our over 80-year-olds, but inversions do exactly the same thing. They also increase with age, significantly so. And then on the right, you also see that smoking influences not only translocation frequencies, but also inversion frequencies in blue. So they're significantly increased with smoking. The other thing that was pretty interesting is that we also looked at telomere length in these individuals. And that was because just really not so much as a marker of aging, which you can actually use telomere length for, but also as a marker of exposure, of environmental exposures. Air pollution has been one, radiation exposure has been another one. But one of the more interesting things was that, in fact, here you see our non-smokers on the left-hand side, and then the smokers with the straight bars. And you see that, in fact, in our unexposed veterans, that the non-smokers had longer telomeres than our age-matched veterans, and then the exposed veterans, it pretty much evened out. So bottom line of this was just that, smoking a pack a day for most of your life was as bad as being exposed to radioactive fallout. So there's a lesson to be learned there. And we also used telomeres in another way, and that was again related to our strand-specific strategies. And we could then use telomeres to kind of as a reference point and help us identify not only whether or not we were looking at terminal inversions or terminal SCEs, and really gave us a good reference point for identifying and really quantifying some of these events, because again, we were all in all new territory as far as quantifying inversions in these individuals. So as I mentioned, we did in fact confirm that inversions occurred at a higher background rate than translocations. And the findings were really consistent with some of our in vitro studies and in vitro calibration curves and just in vitro experiments that we had done. And I think one of the more important things is that not only chromosome translocations, but also inversions really gave a very consistent and improved dose estimate. What kind of dose did these individuals get, which was really the point of the study? How could we improve dose estimates for these individuals, because they really didn't have very good dosimetry done at the time. And we can go back now more than 60 years after exposure and include inversions in those estimates and really improve our dose estimation. So that was the bottom line of that study. I'll just finish up with some of another radiation environment, of that being of space, and how we were able to use DGH and this identifying chromosomes related to that radiation exposure as biomarkers. And I'll start with the twin study, which I imagine many of you might be familiar with, but looking at Scott and Mark Kelly identical twin astronauts, and really working with this team of investigations from around the country for the most comprehensive study of human health effects in astronauts that really has ever been conducted. And it was just really a marvelous opportunity for us to look at chromosomes. And so not only did we, of course, we're interested in telomeres, but we also did the DGH analysis to look at chromosome aberrations in both the Earth twin. So that would be Mark Kelly, who spent the year before, the year during, and the year after the 1-year mission on the ground. And then space twin, Scott Kelly, who spent the year on board the International Space Station. And so when we look at this, so we have Earth twin here, Mark Kelly in green, and space twin in blue. And you can see our pre-flight, post-flight values for Mark Kelly, both inversions in the solid bars and translocations in the stripe bars. And then when we look at Scott Kelly, our space twin, again, the same kind of thing where we're looking at pre-flight, but during flight and post-flight. And you can see, let's look at translocations first. In the stripe bars, you can see that in fact, during space flight, translocation frequencies were indeed increased as you would expect because of the radiation exposure, but then they also start to decrease. This study went out about 9 months after he returned to Earth. So we are losing translocations, but definitely exactly what we would expect from translocations. If we look at the solid blue bars, so these would be the inversion frequencies. And you can see again that they increase during flight, but they persist after space flight, which was a bit of a surprise, but also I think an important observation when we think about the long-term health effects, that a lot of these small rearrangements, like small inversions, really don't kill cells. That these cells survive and they're going to be maintained in the blood, and could be really suggestive of an ongoing kind of instability or damage to stem cells that could have much more long-term kind of health implications. So we were very anxious. We were also really fortunate to have a separate cohort of astronauts, 10 astronauts on shorter duration missions, about 6 months, and their age and sex match controlled. So we were able to do our chromosome and telomere analyses on this larger cohort. And what I'm showing you here are all of the astronauts combined. And on the bottom here are our inversion frequencies. So the same thing, pre-flight, during flight, post-flight. And you can again see that this trend definitely holds up when we add additional astronauts, that there's a significant increase in inversions during space flight, and they're maintained. They decrease some, but they really are persistent out to 9 months to a year post return to Earth. It was also very interesting that we saw this very flight-specific increase in what are called satellite associations, and I'll show you what those look like. But those are also have been associated with low-dose radiation exposure in, for example, hospital workers. So that was another really informative clue, or just supported the idea of these things being related to radiation exposure. I'll just also draw your attention to the top panel here, where we also did quantify sister chromatid exchange, both all along the chromosome and in the subtelomeres, and saw some increases in those as well. But what I'd really like to highlight is this heterogeneous sister chromatids, which was very, very flight specific. And I'll show you again what that looked like. And just here we see these just very uneven telomere signals at the end of a chromosome, so a heterogeneous telomere length. And what was important about that is that has been established as a hallmark of this alternative lengthening of telomeres, and as a result of chronic oxidative stress. So again, another clue that we've gotten from our cytogenetic or cell-by-cell analysis that really points to radiation exposure, because you get increased chronic oxidative stress with radiation exposure. So that really helps support that, as well as these satellite associations. And that's what you see here on the right, which again, as I mentioned, has been a biomarker of chronic low dose radiation exposure. So chromosomes can really be very, very informative, and they've given us a lot of mechanistic insight into some of the changes that we've seen. This was an interesting one here where we used our chromosome aberrations, inversions along the top here, as a proxy for dose. And that's because you can see here that in the astronauts, as lifetime dose increased, the frequencies of inversions also increased exactly as you would expect. And if you use that then for a proxy for dose with increasing inversions or increasing dose, we also see this very consistent decrease in white blood cell counts. And so that's just again, an interesting observation that in this case can be related to radiation exposure. And we also saw that in a completely unrelated study where we looked at prostate cancer patients who were undergoing radiation therapy. And we saw exactly the same thing when we were using either dicentrics or inversions as proxies for dose, that with increasing dose, you see decreases in these white blood cell counts. And again, that can be a very important thing for astronauts on these long duration missions and thinking about their health and performance during a long duration mission. So I'll very quickly wrap up here and just say that, really chromosomes in space have given us a lot of mechanistic insight into the space radiation environment and what that might be doing to, or the implications of what that might be for astronaut health, not only during a mission, but also once they come back. And I think that's all related to these persistent DNA damage responses that we're seeing, particularly inversions and space satellite or space satellite associations that are signatures of space radiation exposures. And the rest of this is really related to some of the other things that support that were also found in the twin study that really have given us a much more complete idea of what's going on. I'll end by thanking, of course, NASA for their funding, but also the different groups associated with the twin study and the space related studies that I showed you, and also just acknowledge all of the different individuals, both here at CSU and at NASA, but also all of the folks at KromaTiD over the years that have been instrumental in developing DGH and making the studies I showed you today possible. So with that, I'll stop and be happy to take questions.
So, yeah, thank you very much, Susan. That was fantastic. And so before we jump into the questions, I thought I would just kind of summarize where we stand today. KromaTiD was founded actually to commercialize directional genomic hybridization. And over the last decade, we have launched a number of products and services. And then we include within that some of the old school techniques that we talked about at the beginning here, including G-banding and our own version of FISH, which we call pinpoint FISH. Pinpoint FISH is unique in the fact that it does use the DGH probes for doing interphase FISH. And by doing that, we end up with an order of magnitude higher resolution than you can get with a back-based assay. But the real stars of the show are DGH, directional genomic hybridization products and services that we offer. And all of the things I'm about to say, you can get them from us as a service where you send samples to us. And we are now commercializing kits. So you can get DGH Insight, for instance, for CAR-T in kit form. And we are in the middle of beta testing DGH Screen with various labs, including the UTMB lab, Michael Kornforth's lab that Susan mentioned in the acknowledgements, but several others around the world right now. So just kind of starting from the left to the right, DGH Screen, that is our whole genome unbiased method. It's available for the human genome. And, Susan mentioned the original version of DGH Screen used 17,000 FISH probes basically for chromosome 3. Chromosome 3 today has 500,000 FISH probes on that one chromosome. It covers all the unique sequence within the genome. There's more than 10 million probes in the entire genome for Screen. Discover is a banded version of Screen. It uses exactly the same probes, but they are multicolored, so it's parallels to M-band. for that. And that allows us to see breakpoints and identify that. And kind of unique with Discover is we know all of the gene content below every single band. And so we use Discover for biomarker discovery pretty routinely these days. And so also available as a service if any of you have any needs for that. And then Insight is localized. It's still an unbiased look, but it's very regional. And so we often use this for gene and cell therapy projects where we can use Insight probes to target transgenes that are being inserted into the genome, for instance, or to profile CRISPR edits, as Susan showed on one of the previous slides. And so if you have any need for any of these types of services, we have a question, which I'll jump right into, about what we think about optical genome mapping. And so we love optical genome mapping, honestly. It's a fantastic technique. It's been, instrumental in making the whole human genome project real, and we can integrate with that data. The negative to it, if there is one, is that it's really just based on DNA strands, and you do not get the view of an organized genome that you get with DGH, where those chromosomes really allow us to map out the real locations of things. We don't have to infer. It's, as we like to say, it's a direct genomic technique. And so we'll take some other questions here at this point. Do we have anything else right there?
Well, I have a question.
Sure, go ahead.
So, I know you mentioned, the stem cells and, can you say more on that? Like, have you had a chance to look at stem cells and any exposed people, regardless of how they were exposed?
Yeah, that's a good question. And it's actually one that, we hadn't really thought too much about to start with.
Sure.
But we're currently involved in a study with NASA called CYPHER. And so we're getting additional astronauts. And now, based on what we've learned, in our previous studies, we now are asking that very question. So some of the sample that we get back will be run through flow cytometry, for example, and we're going to look for stem cell markers. But the idea is that we'll probably see, increased numbers of stem cells or progenitor-like cells in the circulation as a result of this decrease in white blood cells. That cells are being killed, they're dying because of the radiation exposure, and you're actually recruiting, you're kind of speeding up, you're accelerating that whole process and getting more stem cells into the circulation. So I do think the stem cells play a big role in this. And
Because they could be damaged as well.
That's right. They could
And then
just
you
be
could
damaged
carry
right
the
off.
rest of your life.
That's
right?
exactly right. And that's really about the only way you could keep them that long. It has to be something that happened in the stem cell.
Because the white cells don't live that
Mm-mm,
long.
no,
so-
the circulation turns over fairly quickly.
Right,
Mhm.
right.
Right, and we're looking at 40+ years.
Yeah,
Right.
mhm.
Yes.
And 60 in some.
Yeah.
Yep.
All right, so another questions come in for Susan. Can you compare CoFish and DGH?
Okay, yeah. So, I mean, CoFish was really the humble beginnings of the strategy, the strand-specific strategy, and how we can make single-stranded target DNA. And then, for the hybridization of single-stranded probes. And that is the basic idea. It's just that the key thing for DGH is that now we want to be able to do unique sequences. That was the real difference. CoFish work great for repeats, where you could just have a single little oligo to like the TTA, GGG of telomeres, and you could light up a whole telomere throughout the whole genome. But with a unique sequence on a unique chromosome, that was the real challenge. So now, and that's what was enabled really by the human genome sequence, and it has done nothing but improve as they've released new versions of that. So that's the difference.
Please continue to submit if you have more questions. Please see still a number of people are on the webinar. We'd love to hear you, or just comments.
Right. One other question here: what's involved in running DGH in my lab? And so I guess I'll address that one. So KromaTiD offers a kit that we sell and provide. It includes the reagent that Susan mentioned that allows you to perform the stripping. And then we will sell you either standard probes. We have a pretty large catalog of probes, an ever-growing catalog of probes that's on our website. But we also do a lot of custom probes as well. So if you have unique targets, we can target any probe, let's say a transgene probe, down to about 1.5 kb size these days. And that limit keeps going down. So don't hesitate to bring us something smaller and we'll give it a shot. And then to run it, you need a cytogenetic microscope. If you want to run all 5 colors, that would be 5 separate channels plus the DAPI channel. And that's really all that's involved. And so, we would love to partner with anybody that's interested in trying this, particularly if you want to join the DGH Screen beta testing program. We would be happy to provide you some free samples and all the tech service that you can imagine in order to get you up and running. And particularly if you've been using MFish or Sky or M-Band, we would very much appreciate the comparison in your hands about how all this works.
Oh, and same with the CAR-T kit. If you want to do a pilot study, we are, happy to work with you, offer some free reagents to have you give it a go.
Yep.
And see what happens. Yeah.
Yep. Okay. Last question. I have a unique target. It's a non-genomic transgene. Can DGH detect it? I think, yeah, I just touched on that briefly, but yes, as long as it's, let's say, greater than 1 kb, we can detect it. Because it's non-genomic, we can detect wherever it lands within the genome. And so if you're doing, let's say, a lentiviral transgene insertion and it's scattered around the genome, we can tell you which chromosomes it's on. If you're using homology-directed repair at a CRISPR cut, we can tell you whether it's on target or off target. And so it actually amounts to a large percentage of our business working with CAR-T customers right now. So.
Let me add, so if someone's doing just standard fish right now, what would be the, in terms of moving to trying out our pinpoint fish probes, for instance, if they felt like, fish was fine, but what would you say is our, the advantage of our pinpoint fish probes over?
So the pinpoint fish probes use the DGH Insight DNA, single-stranded DNA probe. So they are much more precise. They are true genomic probes, so they are synthetic. We know the start-stop address of every one of those probes. So for instance, they're not back probes, they're not double-stranded, they're not just sonicated to generate some kind of distribution of lengths. And because of that, and because of the way we design them, there's no repetitive DNA, there's no COT. They have a much higher resolution, so an order of magnitude better resolution. So I guess you could call that a smaller resolution. We can resolve smaller targets. And they are very easy for us to synthesize. So if you tell us a target, we can, within 3 weeks, make and qualify a new pinpoint fish assay for you. The other advantage then for that is, depending on what samples you have, you can go back and forth with those same probes. So you can run a pinpoint fish assay in interphase, and then you can run a DGH Insight assay in metaphase. And they will translate beautifully between those two systems.
Okay, great. Did you want to say anything about plasmid services that we now offer? Because that's kind of your baby, I think.
Sure. So a lot of our business is in the cell and gene therapy space. We do both CRISPR based editing projects with people. We do programs with people who are filing INDs, and we profile their drug product for them. We're even now offering release testing for batches of drug product. And so you can think a lot about what DGH, G-banding, and pinpoint fish do in those types of systems is measuring the effect of vectors on the human genome, particularly looking for off-target genotoxic structural variants, all of those kind of outcomes from the editing in experiments and the production of drug product. And so to kind of go along with those services of measuring the effect of vectors, we have started to produce vectors, AAV lentiviral plasmids, essentially any plasmid that any of our customer base would like, we can synthesize it, and we can do that all the way up to and including GMP production of plasmids. And so it just kind of closes the circle, make the vectors and look at the effect of the vectors on the cells that they are editing.
So kind of related, actually, with another question just came in.
Yeah, and so Susan emphasized kind of one aspect of our work, which is the radiation biodosimetry, so kind of randomly generated double-strand breaks. A lot of the commercial work that KromaTiD does, though, is in cell and gene therapy, particularly around CRISPR edits. And so we have spent the last 5 years really focused in that area. Our first projects were with Editas in 2017. And so we have looked at essentially all of the different systems. So Cas9, Cas12a, zinc finger nucleases, TALENs, and there are definitely trends for what happens, but in essence, you're directing double strand breaks with everything other than base editing. And those double strand breaks then give you opportunities for misrepair. And so if you have 2 concurrent double strand breaks, you can form a structural variant in your edited system. And so when we measure those, we look at batches of edited cells, we see a distribution of structural variants, translocations, and inversions kind of distributed across the cell population. And the more editing you do concurrently, the more double strand breaks you have concurrently, the more structural variation you will see. So if you're going to do, for instance, a triple knockout, it's not unusual to see, 5 or 7% structural variation in there. And so points out a good reason to maybe do a cloning step and purify away those structural variants and just look at your kind of pure, well-edited cells. But if that's a place that you are working, we have over 100 cell and gene therapy customers that have taken advantage of DGH over the last 5 years, and we'd be happy to work with you on that.
Okay, great.
Any other questions? All right. I think we're at the top of the hour. I think we would all just love to say thank you once more for coming and very much appreciate this. And thanks, Susan, in particular, for joining us today. And we will be back with another webinar towards the end of the year. And so thank you all.
Thank you. Have a good day.